On a custom, on our opponent, esteemed audience, my name is Ngoc, and today I would like to present to you my doctoral dissertation on engineering cellulose nanofibril hydrogels with bioactive phytochemicals, what functional biomaterials. So first, let's take a look at the motivation behind this work. Do you know that every year there are roughly 190 million animals being sacrificed for biomedical research? For purposes like education and training, fundamental studies on disease, drug development and testing, as well as to extract a wide variety of reagents like antibodies, serum, enzyme, hormones, and growth factors. Especially for cell culture work, different biomaterials like collagen, gelatins are extracted from animals to be used as a matrix for cell culture. Here, I would like to draw your attention to matrigel, a special type of animal-derived material which is used extensively in cell culture. So what is it and why do you use so much in biomedical study? So matrigel is a biomaterial extracted from mouse tumor. It is a mixture of biologically active proteins like collagen 4, laminin, and pectin, heparosulfate proteoglycans, and growth factors. And it is currently considered as the gold standard for cell culture, as it replicates the micro-natural environment for different cell lines. So it is used on various studies on stem cell differentiation, tumor assay, wound healing, regenerative medicines. Although matrigel is highly useful for cell culture, it raises certain safety risks as it is derived from xenogenic and tumor origin, meaning it comes from a different species. It also features high batch-to-batch variation and high cost. And finally, it is associated with ethical concerns since the tumor has to be propagated or created in young mice, and they are typically sacrificed after a few weeks to collect the materials. So for these reasons, researchers are looking for more sustainable and ethical options for cell culture. Plant-based hydrogels are more sustainable or potential alternative because they are produced from non-animal resources, so reducing the contamination and safety risks. And also, plants are historically been a important source of bioactive and therapeutic compounds, so they can be used to create a new and functional biomaterials and especially biomimetic materials, meaning that they can mimic the biological materials like proteins. So the main aim of this work is to create a plant-based hydrogels that can be with appropriate performance that can be used as non-animal alternative for biomedical applications. And in this material development pipeline, we first use a novel material design approach to create hydrogels. Then we tested or characterized the mechanical performance of the hydrogels. We studied the structure and property relationship, as well as to test the behavior of the hydrogels in the working environment. And finally, as the target application is for these hydrogels to be used as the matrix for cell culture, we also study the cell material interactions. So plants offer a wide variety of chemicals and materials. And in this work, I use 3 ingredients. So cellulose nanofibrils, CNF, willow bark extract, WBE, and aloe vera gel, or AV. So what are they and why these 3? First of all, cellulose nanofibrils are strong natural fibers separated from root pulp. They have diameters ranging from 5 to 20 nanometers, which is similar to collagen fibers in dimensions. They are also highly hydrophilic, meaning that they can retain a large amount of water within their volume. They also have low toxicity to cells. Willow bark extract is a great source of bioactive compounds like salicin, triandrin, and piscin. These compounds act as the defense mechanism of the willow plant against the attack of disease or bacteria. So these compounds has antioxidant, antibacterial, and anti-inflammatory properties. The final ingredient is aloe vera in the gel. So aloe vera has a thick and viscous gel which contains mainly bioactive polysaccharide, and it is quite famous for its therapeutic effects like wound healing, anti-cancer, or the ability to activate immune system. So each ingredient here can offer a benefit to the final product. Here I created two types of hydrogels by combining cellulose nanofibrils to either willow bark extract or with aloe vera in the gel. So the main design principle here is to combine hard and soft element. So the CNF are stiff fibrils, so they can be used as the reinforcement or the hard element. Willow bark extract on its own contains only small molecules, and when dissolved in water, it becomes somewhat like tea. It has a brownish color to it. And we also use lactase, an enzyme from fungi, to polymerize the small molecule of catechin into a longer chains, and these chains can act as the soft element of the hydrogels. Aloe vera extract, on the other hand, already comprised of long and soft polysaccharide chains. So the combination between CNF and aloe vera extract resulted in a series of white translucent hydrogels, as you can see here. So let's take a look at the mechanical performance of the hydrogels. So the graph here shows the storage moduli, or you can understand it roughly as the stiffness of the hydrogels. And also there's the dry matter content, corresponding dry matter content, represented by the star of the same color. So the left part of the graph shows here the moduli of single component hydrogels, meaning that they only contains either the pure cellular nanofibrils or pure aloe vera extract. While the right part here shows the moduli of dual component hydrogels. So when we look at the bars, orange bars here, these are the moduli of CNF and willow bark hydrogels, and you can see that there's only a slight increase in the moduli compared to the single component hydrogels. So these hydrogels exhibited low strength even with high solid content, and even after the polymerization, so represented by the bars with the diagonal stripe here, you can see that there's only a slight increase in moduli. The reason was because willow bark extract only contains small molecules, and the chain length was not sufficient to create a secondary network. On the other hand, when you look at the moduli of CNF and aloe vera hydrogels, you can see that there's a significant increase in the moduli compared to the single component hydrogels. So these hydrogels exhibited quite high strength and elasticity, and their performance exceeded that of the single component hydrogels of the same dry matter content by multiple times. Here I also included videos of an actual sample from cellulose nanofibril and aloe vera extract. And you can see that they are strong enough, they becomes much stiffer and strong enough to make a freestanding construct ready for the compression test later on. I also poked it with a spatula to demonstrate that they exhibited relatively good elasticity. We also had a few interesting observations on the interactions of these hydrogels with water. So when immersed in water, we observed that we found that the willow bark extract reduced the water swelling, or to be exact, the iron salt present in the willow bark extract reduce this the water swelling, meaning reduce the water absorptions of the hydrogels. And the willow bark extract also improves the flowability of the hydrogels, which makes it easier to extrude them from a syringe or make it better for 3D printing. We also attempted to make dry foams from the hydrogels of CNF and CNF aloe vera extract. And you can see that there's a significant reduction in shrinkage after drying, which is a problematic issue for the pure cellulose nanofibril hydrogels. And the aloe vera extract also enhanced the elasticity of the hydrogels, resulted in a better resistance to pore collapse during drying and better resistance to mechanical failure. From these results, from the observation, we continue to analyze the interaction between the components, and we found that both willow bark extract and aloe vera extract strengthen the CNF network, but in a very different way. So the salt ion in the willow bark, or the potassium ions that you can see here, they created a denser network by reducing the distance between the fibrils, while the phenolic compounds absorb on the surface of the fibrils, acting like lubricants, so they improve the flowability of the hydrogels. Aloe vera, on the other hand, they comprise of soluble and insoluble fractions. So the soluble polysaccharide chain absorb on the surface of the fibrils, CNF fibrils, while the non-soluble brown grands of aloe vera extract, occupying the interfibrillar space. So together they effectively created a secondary network besides CNF, which allows for more effective and efficient load transferring and dissipation. We came to a conclusion that the CNF, the hydrogels performance can be tailored based on a proper selection of components and ratio, and the knowledge about the interaction between the components can provide a basis for future performance optimization. So after fabrication and testing the material's performance, we also investigated another important aspect, which is cell-material interactions. So when talking about cell-material interactions, there are several aspects like toxicity, blood compatibility, cell adhesion, etc. But here we developed the plant-based hydrogels to be used as a matrix for 3D cell culture. In 3D culture, cells are not spreading out on a flat surface, but they are expected to form a cluster or spheroid and behave like they would in tissues or in living body. So previous researcher observed that cells do form spheroid when they are cultured in certain biomaterials and not in others. So there is a hypothesis about the spheroid formation. That is when cell to cell adhesion is stronger than cell to material adhesion, then the cell will form a tight cluster and vice versa. So in this work, we will test this hypothesis. We tested this hypothesis by measuring the cell to cell and cell to material adhesion on two different biomaterials. One is the animal derived matrigel, and the other one is pure cellulose nanofibrils. And see if the cellulose nanofibril, the CNF, can support the spheroid formation or not. So how do we measure the cell to material interactions? We used two techniques based on the atomic force microscopy method, so AFM-based techniques. So in a simple way, we attach either a glass sphere coated with the materials of interest or a single cell onto a cantilever. So this yellow part here, the cantilever is a spring, a very well-defined spring, and when it comes into contact with another materials or another cells, it can be pulled or pushed away by the interaction force. So first, the cantilever and the cells or the glass sphere attached to it together, we will call it a probe. And first, we will put the probe into contact with the cell colony underneath, and then we retracted the probe. The deflection of this probe can be monitored and converted into force, so we can obtain a graph of force and separation distance. So the lowest point of this force distance curve is called the force of adhesion, and all the area under the baseline 0 here is called the work of adhesion or the energy of the adhesion. These are the two main values that we use to test this hypothesis about spheroid formation in matrigel and in cellulose nanofibrils. We tested this hypothesis on HepG2, a liver cancer cell line that is typically used in liver cancer studies or drug development. So here you can see that the cell-to-cell adhesion is higher than cell to matrigel and cell to CNF in both category adhesion force and adhesion energy. So these values suggest that both matrigel and CNF can support the spheroid HepG2 spheroid formation. And these observation agrees with the actual 3D cell culture experiment, as you can see from the fluorescent images here. However, when we take a closer look, we can see that the spheroid in CNF is more tight, while those in matrigels are loose, more like a bunch of grapes, where you can see the border of individual cells. This shows that the cell-to-cell adhesion was promoted in CNF, while in matrigel it was hindered somehow. So to gain a deeper understanding, we also look at the details like tether and jumps in the force curve, and we counted the number of these details or events and their magnitude to obtain a histogram of the binding events occurred in each force distance in each system and their frequency. So to give you a bit of a context, cellular adhesion can happen either via specific or non-specific adhesion. So in specific adhesion, it occurs in a selective manner, and it requires a matching or occur in a lock and key pairing. And it also involves different or important receptors on the same membranes like cathedrins or integrins. So cathedrins mainly regulate cell-to-cell adhesion. While the integrin mainly can recognize and bind with biologically active materials like proteins, and together they will trigger a coordinated response from the cells. Non-specific binding, on the other hand, happens in a non-selective manner, and it takes place like almost everywhere, if favorable. So when we look at the histogram of the binding events occurs in each system, we can see that there's a peak showing up in the HepG2 and matrigel system, as well as in the HepG2 and CNF, and they are in similar range. So CNF do not have any kind of biologically active ligands. So we speculate that only non-specific adhesion can take place here, while the HepG2 and matrigel are mainly occurred in a specific adhesion mode. This analysis demonstrates that CNF, although they doesn't have any kind of biological, doesn't have any kind of ligand there, but they can trigger a biomimetic interactions. So it can, meaning that the interaction of CNF can mimic what happened here in matrigel. More surprisingly, when we monitor the expression of important receptors like integrin, ITGA5, CDH1, and CDH2, we have also an interesting finding here. So the CDH1 and CDH2 regulates the expression of cathedrins and regulates the cell-to-cell adhesion. We can see that there's an increase in the expression of these receptor in cell culture in CNF, while there is a decrease in cells culture in matrigel. So we came to a conclusion that the CNF promotes the cell-to-cell interaction, while the animal-derived nitrogen reduces the interactions somehow. So in conclusion, this dissertation demonstrates that plant-based hydrogels with plant-based hydrogels can be fabricated using plant extract, and their performance can be tailored based on a proper selection of component and ratio. And we can use the knowledge about the interaction between the components for further optimization. The AFM study also demonstrated that the CNF-based hydrogels offer biomimetic interaction with living cells without disturbing crucial cell-to-cell adhesion. And these plant-based hydrogels are potential as more sustainable and ethical alternative to animal-derived material for biomedical applications. Thank you for your attention. And now I would like to ask you, Professor Christine Siveru, appointed by the Aalto University School of Chemical Engineering, to present your observations on this dissertation.
Thank you very much. Ladies and gentlemen, it is a pleasure and an honor to open today's doctoral defense with a few reflections on the broader scientific and societal context of the work we are here to discuss. The field we are addressing today lies in the intersection of material science, biology, and medicine. More specifically, the development and application of hydrogels for biomedical use. Hydrogels are soft, water-rich materials that can mimic key aspects of human tissue, making them uniquely suited for supporting living cells in three-dimensional structures. And this capability opens up important opportunities in modern medicine. One of the most promising is the creation of physiologically relevant 3D cell models, such as spheroids, which allow researchers to study cellular behavior and test pharmaceuticals in vitro under conditions that better reflect the human body. This is not only scientifically valuable, but also contributes to reducing the need for animal testing, like you showed us, an ethical and regulatory priority. Hydrogels are also used or under development for applications including drug delivery, tissue engineering, and wound healing. As such, they represent a key enabling technology for several future-oriented areas in life science and healthcare. However, the potential of hydrogels comes with some challenges. Their performance depends critically on precise control of properties such as stiffness, porosity, and biocompatibility. Today, many widely used hydrogels are based on synthetic polymers or on animal-derived materials, both of which presents limitations, such as lack of biological cues or the risk of immune response. There is therefore a strong motivation to develop bio-based, non-animal and tunable alternatives that are better suited for medical use and scalable production. The doctoral work we will discuss today contributes to this effort by advancing our understanding of how to create biologically relevant 3D environments for cells, hydrogels. So with these opening remarks, I look forward to discussing this with you. I hope I can call you Helmi.
Yes.
Good. I think you have done a great job in this work that you have presented. So I look forward to discuss this with you. So you have already 3 published papers in recognized journals and with high impact factors as well. You have been the first author of 2 of them and have shared first authorship in the 3rd one. So you have already demonstrated high scientific skills just by doing this. And I really also enjoyed to reading the thesis, the summary, which was very easy to follow. You have presented the background very thoroughly, and I like how you presented the papers also in the thesis itself. I have some general questions just to begin with regarding, for example, when you started here at Aalto, you started in a project that was kind of defined to a certain level before you started, I guess. Can you tell me a little bit about how it was to start and what was already given of tasks to you, and what did you decide upon by yourself from the beginning?
I first started this dissertation with the BioNet project. So the project with the hydrogels from CNF and the willow bark extract. The motivation behind that project is also to create a plant-based hydrogels with stronger or better mechanical performance using a novel design approach, which is called double network hydrogels. And so at that point, I think that I thought that the approach was really interesting in the way that it doesn't it didn't comes from the excessive use of cross-linking or chemical to improve the mechanical performance of the hydrogels, but rather using an enzyme, a green chemistry approach to make the longer chains of the hydrogels, a longer chains of catechin. So I think that it was a very potential approach, and so when we started with that project, we found that there's some problem with the chain length of the catechin. So I moved on to exploring other ingredients from plant extract to find a better candidates for the second network. And so I found I started the second project with the aloe vera extract later on. So it is a very important project to get me started with the getting to know also the cellulose nanofibrils and different variety of the cellulose nanofibrils, and later on to the hydrogels with cellulose nanofibril and aloe vera extract. Yep.
So quite much has been done already on hydrogels. So when you started to work on that, was it hard to find knowledge gaps?
I'm sorry, can you?
Knowledge gaps, something that was unexplored, because there is quite a lot of work on hydrogels already before you started.
It is true that there's a lot of work already with the hydrogels, but the main problem with the hydrogels is the mechanical performance of the hydrogels in general are quite poor. They are soft and wet materials, so and which is why that I aiming for to use the novel approach of double network hydrogels to improve the mechanical properties of the hydrogels. Mainly usually they are it is done by chemical cross-linking of synthetic materials, but the synthetic material also have their own challenges in the way that it's the starting materials always like have some kind of safety concerns when the target application is biomedical applications. So I aim to use plant-based extract or plant extract to leverage the benefits of plant extract in a way that it also more biocompatible with the cells in vitro models, and also it's yeah, I think that it's more sustainable alternative to synthetic and
Yeah, absolutely. Absolutely, I agree with you. But I think there is already some work on combinations of CNF and alginate, for example. I have myself been involved in such hydrogels that also show quite similar properties to yours. So you say that you will avoid cross-linking, but what do you mean then by cross-linking?
So the cross-linking means that the creation of the network, 3D network, so three-dimensional network by creating a junction point in the fibrils. For example, in cellular nanofibrils are only linear chains or fibrils. So there's no connection between them, and mostly they created three-dimensional network by physical entanglement. Cross-linking here, I mean that they create a permanent bonds between the fibrils to make a 3D network.
Like covalent linkages, yeah, and not ionic.
Yeah.
Because I think you can have some ionic bonds in your hydrogels as well.
That's true. If it's oxidized with the tempo CNF, then they have carboxylate groups and ready to be cross-linked with calcium chloride. But I think that it is also a potential route that I thought of. But it's when it's cross-linked with ionic agents, when it immersed in water, the concentration of the ionic salt can reduce and resulting in the disintegration of the hydrogels also. So I think that it's non-permanent, so it's reversible also.
But they can be quite strong.
Yes, that's true. Yeah.
Let's see. Another of these general questions is that you have, of course, done much of the work in collaboration with several other researchers with different expertise. So are you very familiar with all the techniques that have been used here, or is it something that somebody else has done?
I am familiar with most of these the techniques that you use here. Even with the cell culture, although I'm not the one who did the cell culture and like gene expression, but I also spent a short amount of time like learning the cell culture techniques and also the biochemical assays to analyze the cell's behavior.
Yeah, so you feel very confident about all these methods and how they have been done, how they have been performed. So you can, you are very skilled, not practically, but theoretically on the methods that you have also, that somebody else has done.
Yeah, I hope so.
So what, I guess you have had to take a lot of choices during your study. So how how has this been done? Have you discussed with several others, primarily with Monica perhaps, or how have you worked? Have you had your own ideas, or have you discussed in groups?
So for the first publication, since I didn't have a background in cellulose nanofibrils at the times, I only learned it from like pulp productions. So and also the willow bark extract is also a new ingredient for me to work on. So I worked with expertise in willow bark extract purification and characterization. So they also have a few previous publication on that. So I mainly focus on not a full chemical analysis of the willow bark extract, but only focus on the ingredients or the bioactive compounds that I'm interested in. And we also planned the whole project with expertise previously worked on the project, like expertise from VTT. So they have they provided the enzyme. They also provided instruction on how much, what is the doses of the lactase enzyme that I should use in doing the polymerization of the willow bark. So from there, I have an overall overview on the pipeline of material development, and I brought that into the second project on the aloe vera extract and CNF hydrogels myself. And at that time, I think that the aloe vera extract was not that well defined. So I worked with Lucas Fleury, who is an expert on NMR. So together we did the analysis, and yeah, most of the later part I think that I discussed with Monica to have an overview on the project and how to proceed with that. Yeah.
So I guess you used the first study to learn all these techniques, and then you applied it more and more by your own in the second study.
Yes.
Yeah. That's normal, so nothing wrong about that. And you have used a lot of different techniques and methods. Which methods do you think have been most useful for you and most challenging to do or to apply?
I think that is most useful and also most challenging to analyze is rheology.
Oh, yeah.
Yeah, because it seems quite simple that you only put the sample there, you did the analysis and you have a graph, but it's quite challenging to really understand or interpret the moduli or the storage moduli, and also because it is also difficult to, and every time there's a variation in sample in the result because of the sample heterogeneity. So
yeah, you have some heterogeneity in CNF samples often.
Yes.
So they can also give you some challenges in the apparatus.
You mean the preparing the materials?
Yeah, when you're using a rheometer, you can have slip effects, for example. Did you experience that?
Yeah, but I think that because before this project, I also had one project on the pulp samples. So they are even more heterogeneous than CNF. So I think that I face more challenges in that project. And here I already have a relatively good understanding about how to prepare and how to make the sample preparations. So the fluctuation in results is less than that project. So,
yeah.
Yeah. I think that before we have to use the serrated.
Yeah,
or the,
you didn't do it here.
Yeah. Here
use
I
serrated?
no. I think that it's easier to use the flat plate plate geometry, and it also have a better area definition of area, so it provide more consistency of the consistent result.
Perhaps you can pay a little attention to the AFM-based force spectroscopy that you have used. You used colloidal probe microscopy and single cell force microscopy. That must have been challenging too.
Oh yeah, I always thought of the AFM as a separated set of challenges, because it is how to say, it's working with materials at a very small scale, and also when you work with cells, they are biological entities, so there's always a very big fluctuation in result. So I don't know if it can show here, but the graph that we collected like more than 1,000 force curve to analyze, and the amount of data is huge. And
Yeah.
yeah,
Because you have a living material and a very delicate measurement.
yes.
So must have been very challenging. So I'm really impressed by those measurements that you have obtained. So that's very good, I would say. So why have you chosen these 3 studies and not some other things to study when you look at CNF-based hydrogels for medical applications? Why did you choose spheroids, for example?
I'm sorry, can you repeat that?
It was a very diffuse question, perhaps. Why have you chosen these 3 studies? The 2 first is about controlling mechanical properties of hydrogels, and the last one to make spheroids. You could have done something else. You could, for example, have studied hydrogels for tissue engineering or...
Yeah, so I think that always the start is different than what you end up with later on. So it is not actually my plan, but the plan is to create the materials, like the material development pipeline that I show you in the presentation. So first, I would like to make hydrogels that there's some elasticity to it. I think I forgot to address that, because I think that CNF and alginase hydrogels are a bit brittle.
Yeah, they can be.
Yeah. So I would like to make a elastic hydrogels that you can apply dynamic compression test. So the target application initially is for them to be used as a matrix for bone tissue engineering. And I have a theory that if you applied dynamic compression test or cyclic compression test, it can induce some kind of response in the cells that helps them to differentiate into bone cells also. So following the pipeline, I also want to test these hydrogels by embedding the cells, actually stem cells, and create a bone tissues from it. But due to the limitation in the time scale, I couldn't include that final project in this thesis. I actually have ongoing projects on testing the cell embedded in aloe vera and CNF hydrogels in the hydrogels to test their responses. The AFM study is it's not really aligned like in a way that you can study the cell behavior, but it also helps to gain an insight into the interaction at the surface between the cells and materials, because If the cell cannot adhere or doesn't have do not have any kind of adhering site, then it can affect also its or their viability and proliferation. So I think that this AFM is the starting point of the cell to material interaction studies pipeline. It cannot. It is not the final point, but it's rather a starting point of studying the cell material interactions. And I think it's also useful to continue this AFM study in studying cell material interaction with different types of cellulose nanofibrils, and when the cellular nanofibril already functionalized with different bioactive compounds. Yeah, so it's only
It's the first step to study the CNF material with cells using that technique. Yeah, because you can have many different kinds of CNFs.
Yes.
You have used two different ones.
Yes.
So I will come a little bit back to that. Now to something different. What would you say are important differences between in vitro and in vivo assessments? What is not captured in an in vitro test?
So in vitro, I think that it is less dynamic than in vivo system, where, for example, when you test some aspect in vivo models, then it's the whole living bodies running operates in operations. So the sample is affected as a whole from the whole body. While in vitro, you have a very well-defined as best that you can do on the in vitro models. So you can decide what the cells are exposed to. And so I think that the variation in result also less than in vitro models compared to in vivo.
Yeah, I think the main, as you also said, the main difference is that you don't have an immune system that interact with your experiment in vivo. The immune system will try to make it as good as it can for this particular animal or a human being. So it will, in a way, reduce the effect compared to if you do it in vitro. So in vitro is a much more sensitive test to because you have no immune system there. And you don't have blood vessels, for example.
Yeah, because for example, like I think the difference also applied in 2D and 3D. So in 2D, the cells are spreading out on a flat surface, and they are exposed to a uniform gradient of like nutrients and metabolites. So why in vivo there's a volume and distant dependent gradient of those. So yeah, and that's true that in vivo there's a strong role of immune system where the plasma cells or component can also decide to reject the implants or the materials.
I have also been part of a study where we saw very clear tendencies in vitro, very, very clear. And when this was done in vivo, there were no clear differences at all. Because the immune system could cope with that negative effect that you had seen in vitro. So I think it is a very important step to go from 2D to 3D, but still you have this limitation in the spheroids that you don't have the immune system, and also it is a question about transportation of nutrients to the cells in the middle of the spheroid, and waste products from the cells out of the spheroids, because that must happen with diffusion, and you can have many cells there. The distance from the solution you have the spheroids in can be too long. Have you some thoughts about that?
Yeah, so I think that it's not challenges, some necessarily challenges, because the spirit is usually used to replicate the tumor environment. And if you have and use for like drug testing, for example, so if you test one specific drugs on a 2D surface, then it will it will likely to result in a false positive response from the cells to the drugs. But the spirit, as it is size dependent, so there's gradients, like you said, on the penetration of
Oxygen
and
yeah, and the drug.
and nutrient and waste metabolisms. So it also reflects a better a tumor.
Yeah,
It's exposed to a drug, and it also result a more realistic result. If you test
compared to 2D, yes. But you will have to consider also the size of the spheroid, because you don't want the cells in the center of the spheroid to die from lack of oxygen. You want them to die from your medication. So you have to have sufficient transport mechanism to the core of the spheroid as well. So they cannot be as big as.
Yeah, there's a limitation on the size. Yeah.
Do you know that limitation?
I think that if it comes above 200 or 100 micrometers,
yeah,
it will develop a necrotic core. So yeah.
They should not have a necrotic core.
I don't know if it's a bad thing or a good thing. If there's a necrotic core, because it depends on what you're looking for. If you want to study the actual progress of tumor, then I think it reflects better the realistic reality of tumors.
I think tumors in the body, they will certainly have blood vessels. They take blood vessels. So it is an issue that you have to be able to control the size of the spheroids, to have good models for the drug testing.
I don't know, because we didn't like really dig into that spirit.
No, probably not. But you were able to make spheroids, but probably not to
control the size.
Probably did not try to control the size, but that could be a next study.
No,
perhaps.
that would be, yeah, for another study. For now, we just analyze if the cells do form a tight spheroid or not. And yeah, we didn't really pay attention to the size control.
No.
No.
Next paper.
Yes.
Yeah, or you say that plant-based materials have inert nature. I think I understand what you're meaning about that, but I don't agree with it. So you say literally that plant-based materials are considered inferior candidates in the field due to their inert nature. And I don't think they are inert. Can you explain why you're saying that they are inert?
Yeah, probably when I wrote it, I thought mainly of cellulose nanofibrils because cellulose are quite inert materials. They don't provoke any kind of biologically response from the cell. So
Yeah, I think that is what you were thinking about. But I don't think cellulose is completely inert either because it has the OH groups. And if you have a tempo oxidized CNF, you have also carboxylic acid. As long as you have functional groups, you don't have a completely inert material. They can react. And yeah, like hydroxy groups, aldehyde, carboxylic acid. So we have all kind of functional groups in plant-based molecules.
it's true.
And also the phytochemicals you are been using, you say they are bioactive compounds. So then they cannot be inert.
Yeah. Yeah, I think that's
I think you agree with me now.
yes. Yeah, I think it's difficult to define the whole plant kingdoms as inert or
Yeah.
bioactive because they contain a lot of different ingredients in there.
Yeah.
Yeah.
Yeah, so let's discuss production of CNFs a little bit. Because you have used two types. It is a mechanically produced CNF that you have not changed chemically. And then you have also done tempo-mediated oxidation as a pre-treatment. Have you done it yourself?
No, in our department, I think that it's produced as following a standard procedure by the technicians.
Mm.
So I learned about the procedure, but I didn't do it myself.
No, but you are familiar with the procedures. So also you say that the energy consumption, it goes down when you do this tempo oxidation first, and that's true, of course. But can you elaborate a little bit about those processes and the cost of them and the products, how similar or different they are?
So in native cellular nanofibrils, native cellular nanofibrils are produced by mechanical fibrillations. So we, after washing them to induce the swelling of the wood pulp, I think that we'll put it into a refiner and a homogenizer. So the wood pulp is like exhibiting shear stress. So they are like separated into a smaller fibrils without any kind of chemical modifications. So they retain the native surface chemistry.
Mm.
But in tempo CNF, you oxidize it with tempo chemicals. So you introduce the carboxylate group onto the C6 positions. So converting the hydroxyl group into carboxylate. So you have more negative charge to the wood pulp, and so it also resulting in repulsion of the wood pulp. And so when you homogenize it with mechanical stress, I think it's the separation happens in a easier way because the wood pulp already like a bit separated from each other instead of like stick together like in native cellular nanofibrils.
But you're using a lot of chemicals, and the product is completely different from the native CNF, as you call it.
Yeah. So I think the final products, the native cellular nanofibrils produced by mechanical fibrillations are more coarse.
Yeah, they are coarse.
The diameters are thicker, while the tempo CNF are more finer. So you can see clearly from the diameters when you observe it with AFM images. And I think that the tempo CNF, the tempo oxidation also like cut short a bit of the native fibers.
Yes, the DP will be lower. But you said that you get carboxylic acids in C6, but you get also another functional group, approximately 20% of the carboxylic acids. Which one is that?
I'm not sure.
No, you get aldehydes. Aldehydes, it's when you do the oxidation, you first form aldehydes, and then most of them is transferred into carboxylic acid. But you will have quite a lot of aldehyde groups left when you finish your oxidation. So unless you are either reducing or oxidize them further, you will have that. And they are also very reactive.
Okay.
They can react very easily with amines, for example, like a click reaction. And maybe you have amines in the matrid gel, for example. I guess you have that.
Yeah.
But you did not use the tempo in the last
No.
study.
No.
So you say you use tempo, but that's only the catalyst. You use some other chemicals as well.
It's bromide.
Sodium bromide, and then the oxidant. What's that? That's hypochlorite, sodium hypochlorite. So if you wish to get rid of the aldehyde groups, you have to either oxidize them with chlorite, which is a stronger oxidant, or you can reduce them to alcohols by using sodium borohydrate, for example. So you can play a little bit around with that chemistry. And you can also, because you have approximately 1000 micromole per gram. Why did you choose exactly that level of charge density?
I actually didn't choose that level. It's as I mentioned, it's produced in a standard procedure by the technician. So they follow the strict dosage of the catalyst and tempo for each batch of their CNF manufacturing. So I only use what they provided me. So we didn't actually work on changing the level of carboxylate content.
But it's quite easy to do it if you wish. You can just use another charge of sodium hypochlorite. But I don't think the level you have used is a bad choice. I think it is a quite good choice. I've used it myself a lot, around 1000. So works very well in biological systems. So now we have discussed the production a little bit, and we will also you have use different solid content of the CNF.
Yeah.
You used 1.2% in the first study and 1% in the second.
Yeah.
Why did you do that? And
it's a funny story. So in the first study, when I first found out that the polymerization of the catechin compounds did not produce in a very stiff hydrogels, I attempted to increase the dry matter content to see if it helps with creating a stiffer hydrogels. But we found out that the problem was the chain length of the catechin was not sufficient. It was not like because of the dry matter content. So in the second study, I use only 1% because it's already like it's more convenient for calculations of the ratio.
okay.
I increase the dry matter content of the aloe vera polysaccharide up to 5%. It's more convenient for calculation, and also it's already sufficient to make a stiffer hydrogels. So I use 1% only. It's not, I don't think it's necessary to go up to like 1.2. It's, yeah.
1% is fine, but they are extremely dependent, the rheology, on the concentration.
Yes, that's true.
So it is so much stiffer and viscous if you use 1.2 compared to 1.0.
Yeah.
It's a dependency in an exponent of 3, approximately.
Yeah. I think that I didn't show the actual sample of aloe vera, the CNF and aloe vera hydrogels of like 5%. It's super thick already. So I don't think it's like really necessary to go up to like 1.2. And also the raw tempo CNF that I received from the technicians also only have like 1.6 weight% or something like that. So we try to use like a compromise level. Yeah.
Yeah. You haven't determined the carbohydrate composition, have you? The hemicellulose content and type.
I think I did, but not reported in the publication. So it's like roughly like 21 to 23% in tempos, or it's quite long ago, so I don't really remember.
No. And what is the main hemicellulose? You have used birch. Have you used only birch?
Yeah.
Yeah.
Only birch. I think it's mainly cellulose.
Yeah, it's mainly cellulose. But I don't know what kind of wood it was in the last study, because then you used a commercial sample. And I'm not sure if you know what kind of wood they are using.
No, we didn't like conducted a full chemical analysis of the sample because of the CNF, because it was obtained commercially from UPM.
Yeah,
So and the main focus is like more on the cell material interaction. So we didn't like characterize the material ourselves, and also the amount of sample that we have from UPM is like really small.
Okay.
So we only have enough to functionalize the glass sphere for AFM study. Yeah.
I don't know if they are giving up the details in their products, keeping their details.
I don't know, since it was purchased from by University of Helsinki. So
And from the willow bark extract, you have used the low molecular weight compounds that you got out from that extract. And I think the main interest was in catechin that can be polymerized.
Yeah.
So this was the component that you wanted to have most of, or were most interested in.
Yes.
And you have analyzed it, and I think you got found out that you had about 2%.
Yeah.
So very low amount. And those low amounts, you also have quite low amounts of the other components there. Like polythene, you have 1%.
Yeah.
And then you say that you have a bit more of triandine.
Yeah, 300 and PCN also.
Yeah.
So.
But you didn't find standards for those.
Yeah.
So you could not determine them exactly.
Yeah, so I use HPLC and like external standard.
Yeah.
So we need to have pure 300 and PCN for that, and it is very difficult to obtain. So I skipped the quantitative analysis of 300 content in the willow bark because we are more interested in salicin and in catechins especially.
Yeah. But then you assume that you could have perhaps 12% of triandine based on others' published work.
Yeah.
And if you say that, and also you have what you have determined, which is a little bit of salt, and then you account for approximately 15% of the content in the willow bark extract.
I'm sorry, I didn't catch the last part.
That you have accounted for approximately 15% of the whole batch of willow bark extract, or is it something that I have missed or overseen?
You mean the salt?
You have a little bit of salt, you have a little bit of catechin, 2%, you have approximately 12%, or you assume that you have 12% triandine. And altogether, this is approximately 15% or something like that. Yeah. This means that you have around 85% that you have not analyzed. What's that?
So based on the previous study, I think there's only trehalose, other compounds like trehalose or tannins also in the willow bark extract, and we base mostly this study on that previous chemical analysis of willow bark. So at first we expected to have like 11% of catechin, and then when we raise the dry matter content to 10% or 12%, then it will have some effect, but it's like too low because the content of the willow bark extract also vary depending on the source of the willow bark and the harvesting time. So yeah, so in this work I mainly focus on material development, so I didn't want to focus on the chemical analysis of something that already
You have used you have adsorbed onto your fibrils a material where you don't know what 85% of it is.
Yeah, unfortunately, yes.
So what do you think it is? Carbohydrates, perhaps small monosaccharides, tannins, something like that.
Yeah, I think
you used hot water extraction.
Yeah. So I guess that mainly it's some sugar, low mono or D or three saccharides, and then there's some tannins also.
Mm.
Yeah.
Yeah, because they are probably water soluble since you used hot water extraction. But you got quite little of what you really wanted to have, catechin. Could you have done the extraction differently to get out more of it?
I think that based on the previous studies, the temperature and the procedure was already optimized to gain the highest amount of extracts from the willow bark. So I just used the same procedure.
Are you sure if it was optimal? Because I would guess that it would have been better, or that you would have gotten a higher yield of catechin if you had used organic solvent like acetone, for example, or a combination of acetone and water.
Yeah, I think that there are several studies that use a series of different solvents like acetone or like ethyl
Or you can mix.
acetate or something to increase the amount. But since the aim here is to use the hot water extract to reduce the presence of solvent or other chemicals in the hydrogels for biomedical application. So that's why I use this specific procedure.
Yeah,
But
you
yeah,
had
it
the
can
procedure,
be.
but if you were about to do it again, or at least if I should have done it, I would have tried to mix water and acetone, because you vapor away all the solvents afterwards, so you will get rid of those, and then you will get a fraction richer in catechin.
Yeah.
Sure about that?
Yeah, probably it's going to be.
It has a phenolic group also, so a mixture would have taken better care of both the more hydrophobic part of the molecule and the hydrophilic part. I assume that. At least I would have tried it. So the reason why you got actually not the polymer out of catechin, but an oligomer with DP around 7 was because you had too little of it. Did you try this polymerization isolated with only the pure catechin?
Yeah, I did, but the result of the polymerization is also quite low. So we assume, I think it's roughly around 2000 or something. So it's a bit higher than the willow bark extract, polymerized willow bark extract, but still quite low. So I assume that it's because of the lactase strain enzyme was not that sufficient.
Okay.
Or the condition of the polymerization was not that favorable for making long chains. Yeah.
No. Mm. So you have, in this first paper, you have assessed the adsorption by using QCMT, which is a very good technique. So perhaps you can explain figure 4 in the paper. I don't know if you can have it on the screen.
Mm.
Yeah. This one, exactly. So this figure C, can you explain what's happening there with all those curves and how they go up and down? Or you can start with A.
So in this part, crystal microbalance dissipation monitor. So there's a crystal, quartz crystal coated with a gold surface, and we already coated this sensor with a layer of CNF or tempo CNF, and then we inject a solution of the willow extract into it, and the vibration of this quartz crystal depends on the amount of the absorption of compounds onto this CNF layer will result in a change of the vibration frequency, as well as the dissipation, like the decaying of the signal, depending on the amount of material absorbed on that sensor. So here, we will soak in, inject it with water to stabilize the vibration of that quartz crystal.
So perhaps you can point where you have this stabilization period.
Yeah.
It's
So
there.
here,
Yes.
before this point, I already stabilized it for a couple of hours to make the baseline as stable as possible. Then we inject the willow bark solution in, and it resulted in a change in frequency, indicating that there's some material absorbed on that cellulose nanofibril layer. And the blue line represents the absorption on tempo CNF, and the brown line represented the absorption of the materials onto the native CNF. And then we rinse the crystal, we rinse the sensor with water. So it resulted in some resorption of the materials onto the surface. And this is what we have in final, the final point, indicating the amount of material remains on the CNF surface.
So you get a little more absorbed on the tempo CNF.
Yes.
Why do you think that's happening?
So we think that there's the tempo CNF are more fine, so the diameters are smaller. So we simply have more surface.
Yeah, you have a larger specific surface there.
Yeah.
Yes. Sure, that's natural.
And we also conducted a similar experiments on the catechin, pure catechin, and the same thing happens in in a way that the tempo CNF allows for more absorption, volume or absorption than the native CNF.
And then you come to the curve C.
Okay, so in the C graph, we have the changes in the frequency, meaning that the amount of the material absorbed on the sensor, while the changes in dissipation indicates the viscoelastic property of that layer. So if it's increased more, then it means it's softer. While if the delta D is smaller, indicates a stiffer surface.
Yeah. And then you see that at least three of those curves, they go a little bit down first. What's happening? You go down before I go.
Yes, so this means that when the first absorption takes place, then it's becomes stiffer.
So it's a solidification in the beginning.
Yeah,
And what do you think is happening regarding?
I think there's
Regarding-
some release of the water.
Yeah, so that is the mechanism and the proof of that this absorption is entropy driven.
So when the materials absorb on the surface, it releases more water and increase the entropy of the system. So it's more favorable and it's entropy driven process.
What about hydrogen bonds?
I think that when the materials or the small compounds absorb on the surface because of the entropy driven entropy increase, then the materials comes close enough to the CNF. And I think that the hydrogen bonding can be established when they come close enough because hydrogen bonds are short range.
Yeah, so they are there as well, but they are also in the water phase.
Yeah. But like, which one is the driven force?
You say, and I think it is a more and more understanding in the scientific field now that the absorption processes are driven by an entropy effect, and that is that water can be released from the surface, the water that was there before you had these absorptions into the system, those small molecules into the system. When they come into the system, they stuck on the surface and they stay there and do that because then the water can go into the big water phase. So that's discussed and debated.
I think the hydrogen bond is what kept these materials left. Exactly.
Perhaps, yes, they're still there. Yes, and the rheology curves. Rheology is difficult, and perhaps you can show the curve. I think it is figure 6, where you have this overshooting effect. And yeah. So there the overshooting effect, can you point at it?
This
Yeah?
or this?
Yes. So what does that mean from a rheological point of view, that you suddenly get an increase in the storage modulus?
So when the stress in the system or like become too high, so the fibers are pressed together, then there's a critical point that there's block formation.
Mm.
So there's an internal structure that resists the flowing of the system. So there's the overshooting phenomenon. It represents that the shear becomes critical, so that it resists there's some structure like flux form that resists the flowing of the system. And when we add the roller bar in, we observe that there's the overshooting disappear. So meaning that the CNF fibers flows quite easily, even at that critical shear strength.
So you get more this overshooting effect. That indicates that you get more resistance in your system.
Yeah.
And that resistance is reduced by adding more and more of the extracts. Yeah, that seems like a good explanation. Your swelling test, they indicate that for the CNFs, the surface was covered already at the lowest level of willow bark extract for the native CNFs. But how was this for the tempo CNFs?
I'm sorry. Can you repeat the question?
Yes, when you did the swelling tests.
Yes.
For the native CNFs, it looked like the surface was covered already by the absorbents at the lowest level of willow bark extracts, but I don't think it was like that for tempo CNFs.
So in this swelling,
that it was covered already from the lowest level of addition.
yeah. So so the swelling reduced the swelling ratio already reduced at the lowest amount of willow bark added. So we think that it's because CNF are quite sensitive with the presence of salt.
Yeah.
And so when it's already at the lowest amount of willow bark extract added, it's already reached the critical level to create a denser network, so reduce the repulsion between the fibrils. So it already reduced this water swelling even at the lowest content of water willow bark extract.
For the native CNF, but that's not so very much charged. It has a very low charge.
Yeah. So
but the tempo, on the other hand, has a very high charge and much higher specific surface area as well.
yeah, so I think that there's more charge
Yeah.
Available for the willow bark, for the salt ion to neutralize in a way. So it takes more willow bark to reduce the repulsion between the tempo CNF fibrils.
Yeah, you have two effects. You have the salt and you have the additives that have to cover the surface. So since you have a larger surface, it will need more material.
Yeah.
You say on page 9 in this first publication that you have more and stronger contact points to the absorption. So I agree with that, but can you make a drawing of how you think the contact area looks like? What kind of bonds do you have there? And for the tempo, for example, where you have a lot of charges, you have hydroxyl groups, and you have some adsorbed material. And how do you think those contact points will look like?
You mean for the tempo CNF only?
Yeah, you can. Yeah, that's more complicated. So.
Mean that maybe the willow bark extract also shield the repulsion between the fibers so that they can create a more physical entanglement, like locking of the fibers together with the help of like willow bark extract.
Yeah, but I'm thinking about only the surface area between two fibrils that have this adsorbents on the surface. How will it look like? How are the bonds there? And the interactions is not a straightforward question because it will be many. I'm not sure if I were able to, I'm able to draw it myself. So, but for sure you will have hydrogen bonds.
Yeah, but yeah, I think that it looks like tempo CNF. It will establish hydrogen bonding with the phenolic compounds at the hydrophilic area, and then it will also have the hydrophobic area.
So how will that look like in that contact area? Will they perhaps, I think they will try to come in contact with a similar compound on a neighboring CNF. It's like similar compounds, they like to stay together.
So maybe the glucoside side absorb on the surface and establish hydrogen bonds, and then the other hydrophobic part is attached to the hydrophobic plants of the
Something like that. I imagine something like that.
Yeah.
And then you have hydrogen bonds as well in those areas that are not completely covered.
Yeah.
Yeah, you were able to control the swelling of these hydrogels by adsorption quite well, but hemolysis test did not. It was not that positive. So, is that the reason why you chose not to continue with that gel?
Yeah, exactly. Because we did the hemolysis assay to test to have a preliminary view of the biocompatibility of these hydrogels with cells, and we also tested with standard compounds like salicin and catechin. And I think that it's not because the result shows that it's not because of those compounds, but more likely because of the whole extract somehow. I think that it's because of the high content of salt. It also created a imbalance in the osmotic effect that causing the lysis of the red blood cells.
So you think that the salt is a component to blame here?
I think that it's the whole willow bark extract as a whole, because it contains too many low molecular weight, and it can create a pressure due to the osmotic effect on the red blood cells. So I think that if we're plan to continue, for example, if I want to continue with this willow bark extract, I'll have to purify the willow bark extract to obtain only like catechin or the salicin, so that it's and create less effect on the osmotic. Yeah.
Yeah. Have you thought about trying to get rid of the salt by dialysis, for example?
I did dialysis for the aloe vera extract because it contains like large molecules and then small salt ions. But for willow bark, the average molecular weight is too small, so I don't think that dialysis would work on to remove the salt ions. Yeah.
No, probably not. I'll just start up this machine. Yes, so let's continue with publication 2, which I think is quite straightforward. You have combined tempo CNFs with acemanan. And I think you mentioned it, but perhaps you can describe what is the role of acemanan in the aloe vera plant?
So the acemanan is a storage polysaccharide in the inner gel of the aloe vera, of aloe vera, because it can retain a large amount of water, and it helps to contribute to the plant's resistance to drought in dry climates. So it helps to retain larger amount of water, I think, compared to CNF.
Yeah. Do you think that's strange when you look at chemical composition compared to tempo? Do you think it is surprising that it can hold even more water?
Yeah, I think that it's if you look at the chemistry, then it's quite similar, because they are both polysaccharide, but I think that the acemanan is less crystallized compared to the CNF, because CNF is more crystallized due to the production photosynthesis of the plant also. So they also have the inherent crystallization, a large crystallization. For the aloe vera acemanan, I think that it's mainly used as the storage polysaccharide, so they are more freely move around in the gels to be ready to be metabolized by the plant for, yeah, so.
And there are several big differences between them. As you said, it is the crystalline part that you have in CNFs, but what about the charge, the surface charge?
Tempo has a higher charge, higher negative charge compared to the aloe vera acemanan.
Yeah, it's not charged. So that should, what should that indicate regarding keeping water, holding water?
So I think that it should have less.
Yeah, but it hasn't.
No. So I think that it's because of the solubility of the, no, like I mean that the chains have more surface area compared to
I think so, because this is a very important point. Because the tempofibrils, they are not separate polymers that can freely move in the, they are bundled together, and the charge is only on the surface. So we have so many more surfaces in the solution for acemanan compared to tempo. They are stiff bundles of cellulose molecules with charge on the surface. But compared to acemanan, most of the polymers are in the fibers, even though they are thin. They are thin, but there are a big difference between fibers and polymers that are dissolved. Yes. And there is also another thing that is very different. That's the composition. So cellulose is very special about these glucose monomers that are bonded together with this 1,4 bond. Yeah. And here you have much more irregularity, even it's not that irregular, but it's different, more irregular, not that stiff. And they don't form the bundles.
So for acemanan, I think that it's both 1 to 4 and 1 to 6 linkage. So
much more freely.
yeah.
So that also accounts for many of the more flexible properties that you get when you mix the two. You have freeze-dried the gels and looked at them in microscope. Can you describe what's happening when you are freezing gel and then freeze-dry it afterwards or dry it afterwards?
So I freeze the so in a way that freezing is immobilize the water molecules in the system, and then in the freeze drying is like lower the pressure, so the solid, the water molecule in solid state can like subline into air, like gases state right away, so it doesn't go through the melting point.
No.
Yeah, so
then you have solidified the network before you remove the water. But what happens when the water freezes? What happens with the polymers and the fibrils when the water freezes?
So the water crystallites and create and it changes the structure of the polymeric network.
Yeah.
Yeah.
How?
I think that it's also expanding the pore because the frozen ice crystal are larger than the normal water molecules in liquid state.
What's happening is that when you start to freeze, you first have these very, very small ice crystals, and that's only water. So all the polymers there are expelled out as the crystal also grows. So then the solid content of the polymer and the CNF increases a lot between all the ice crystals. So then you are forming a porous structure that you did not have before. So you change the structure of your hydrogel completely when you are freeze drying it. So and it's not certain that you get back to the original one if you immerse it in water again, because many of those bonds that you get between fibrils, the hydrogen bonds, they are so strong that water will not open them up again. So you can actually not compare completely a freeze-dried gel with what you have before you freeze it.
Yeah.
So in a way, you induce a lot of bonds between polymers and fibrils, like it's kind of cross-linking too. You get a new structure. If it is a highly charged tempo sample, it might go almost back to how it was before the freezing when you soak it in water long enough. But not for native CNF. This is the effect that we call cornification in pulp and paper chemistry. What do you think if you brought cells into those very solid gels?
When I did the experiments with the cells, I mixed it directly with the hydrogels because I think it's better. Like they can be like extruded and with the syringe, so it's allowed for mixing with the cell suspension when it's still in the liquid hydrogels, or like it's still in the wet hydrogels. Yeah, this drying process is just to test if it's possible to dispense the cells later on and use them as the dry construct and seep the cell on top.
But if you don't dry them and use the gels as they were originally and add cells into those gels that are very stiff, how would it go with the cells? Will they like this environment?
Um, I imagine that the hydrogels or the dry sponge can absorb water and become softer.
Yeah, but we don't think about the dry samples. But before you dried it, before you dry them, they are quite stiff gels.
It still can be mixed with the cell suspension. This stiff construct was only for compression test to test the mechanical the moduli of the hydrogels, but I didn't intend to seat the cell directly on these stiff constructs.
No, because probably they would not survive. That's my guess, because they are so stiff. So diffusion in very stiff gels will be very restricted, I assume.
Yeah.
Don't you think so too?
So I mean that if the mesh size is still large enough, then the cells can like penetrate into the inner core of the hydrogels. But that is something that we have to test in that setting. In my case, I stop at already the ratio of 1 to 5, and at that level, the gel is still like manageable, like mixable with the cell suspension to create a hydrogels with the cells embedded in it. And I think that we tested and the cells are alive, but in the long term, the gels swell.
And then they are not that stiff.
Yeah.
Later on, but if they are so stiff, I guess diffusion of oxygen, for example, will be a limitation for the cells. And also, if you try to 3D print a very high viscous solution with cells, I think you have tried that or yourself, or
Yeah.
with bioactive glass, for example, you can get very, very high viscosity also.
And if it becomes too high, then it's the pressure required to for 3D printing become too high, and it might affect the cell viability or survival rate also.
Yeah, so it kills the cells.
Yeah.
In the printing process. You suggest also in this case that when atom analysis is absorbed on the surface, the mechanism is entropy driven. But I don't think you have an experimental evidence for that from the QCMD in this case.
So the QCMD. . Um,
Write it somewhere that you cannot see it in QCM, but you're still sure that about that it is like that, and maybe it's like that.
yeah, I wrote it that the main driving force was the increase in the entropy of the
Yeah.
system.
Yeah, but you don't have that curve that you had for the below birch extract, where you could actually see it from the QCMD data. You cannot see it here.
Yeah, I think that it's might also happen here that the water was released from the surface of CNF, but it's just the increase in the mass of the from the absorption of the polysaccharide chains like outweigh already like
Yeah. So they cover this effect, so you cannot see it.
I guess so.
That might be. You claim in this article that no other studies show similar enhancements as in your case. Don't you think that's a very bold statement?
I mean, I think that the enhancement applies for the aloe vera and CNF and aloe vera extract because I saw also another articles working on tempo CNF and aloe vera extract, and the result is a decrease in the mechanical performance. So that's, I mean, compared to
Yeah.
yeah.
But for example, if you mix CNF and alginate, you can see similar effects. I have an article I can give to you afterwards.
Yeah.
I don't know if it is equally good as this one, but yeah, can be a bit difficult to say things like that because it might be that somebody has shown something equally good that you have not seen.
Yeah.
Now to publication number 3. So I think this article, as all of them, is also very well written. Very easy to read and with clear aims and a logical flow from introduction to conclusion. And here you have done this very impressive work with AFM. And you have chosen 2 different cell lines, one which is connected or is a kind of cancer cell line, and the other pluripotent stem cells. So it's a bit 2 completely different kind of cells.
Yeah, we wanted to test on 2 different cell lines. On HepG2 is where the spheroid model are used extensively for drug testing. The other one is like pluripotent stem cell because they are relied more on hydrogels in their cell culture practice. So we wanted to see if this hypothesis applied to both 2 different cell lines. So that's why we also included the stem cell in that study.
Mm-hmm. And you had this hypothesis that the stronger cell-cell versus cell matrix adhesion would determine this spheroid formation. But your result showed that it was not particularly obvious that it was like that. So I like the way that you are setting up a hypothesis, tests it, and when you see that the results weren't quite like what you expected, you are digging into what's the reason for that. But I'm wondering about several things. For example, what happens with the cellulose nanofibrils in the samples with the spheroids? Where are they? Are they inside the spheroids, or are they expelled out of the spheroids?
I guess that it becomes either can be pushed away or making the space for the cells to stick together, or they can be like embedded into the between this cell to cell space, extracellular space. But because in these fluorescent images, the magnitude hours, it's too small to see the individual fibrils.
Yeah, you don't see it from the images. No. And there are no other ways that you can assess this. Where they are.
I guess if you really want to see it, then there's a way that you can freeze use a cryo SEM or a TEM to make the cross section of the cells, and then you can see it under the SEM or TEM.
Yeah, maybe. I'm wondering why you didn't also include, or first, why do you make, you can now make spheroids without any biomaterial. So what's the reason to introduce a biomaterial to make spheroids? You could have done it without.
Yeah, that's true, because typically spheroids are produced by, like, let's say, deny the cells of attachment site. But I think that there's some studies saying, claiming that if you include the biomaterials as a adhering site for the cells, then they also preventing the abnormality in the gene expression and cell functionality. So that's why we're aiming to also include the biomaterials into the spheroid culture experiments to support them better.
Yeah. And I also read that you can mimic the extracellular matrix in a way so that the spheroids have a more natural environment around them. So we can also differentiate this extracellular matrix. For example, if you use it for stem cells so that they can develop differently.
Yeah.
That could be a reason to get more physiologically relevant environment. You write that from literature, you have the cancer cells, they form spheroids in 0.8%, 1%, and 1.7% CNF hydrogels. But you have chosen 0.5 as the concentration of the CNF hydrogels. Why did you use 0.5%?
So I think that it's a standard procedure that has been established in the University of Helsinki labs. So they use mainly that. concentration and also to match with the gel concentration so that they can be compared.
So that's the same concentration? Because it's
resulting in the same stiffness of the hydrogels.
Okay, it's not completely the same stiffness because you say that it's on similar level, but they are quite different. They are. It is a big difference in the mechanical properties between the matrigel but you're referring to literature here because you based on literature values, you say that the CNF at 0.5% concentration have a certain viscosity and storage modulus. And then you compare that to matrigel, which is actually quite much higher. So they are not completely comparable. If they should be, you should have used higher concentration of CNF. Then they might be more similar.
Yeah, I think that, yeah, I think that we proceed with the cell culture first. And so it's, it was later on realized that this is a problem, but I think that, yeah.
You could also have measured the values instead of referring to literature. Little bit about the size. I think they are about 100 micrometers, are they? And varying from 20 to 100 or?
To 100 or 115 micrometers. So as you can see, we didn't really control the size of the hydrogels.
No. Yeah, so I think that could be something to try later on.
Yeah.
To secure that they get sufficient oxygen and nutrients. All in all, I think it was a very impressive work. My job is to find the weak points. So I have some final questions. We have actually spent, I think, more than 2 hours now. So I have one question first. What about no, first, do you think you have obtained the primary objective of the thesis? Have you fulfilled your plans? Have achieved your goals? Have you? Do you think so?
I think that for me personally, I think that I made a hydrogels with tunable mechanical properties from plant extracts. But to make a true plant-based alternative for matrigel, I think that we still have a lot of work to do because we need to test these hydrogels in the actual cell culture settings, and we need to observe them in a more dynamic environment, like in the culture medium instead of in water, and like
yeah.
So I think that it's still in progress work.
Yeah. Yes, it's a huge field, a lot of things to do. But if you were about to start the whole study now when you know all the problems that you have had, but you have done something differently.
Um, I think that I will do more, not like do it differently, because everything like some experience to learn and a lesson to learn from. So from now, if I continue my work, then I will, I say, develop it a bit more into the process, into the direction that I can test them in a actual working environment and studying those aspects that I haven't been able to cover in those previous publications.
So you would have done it the same way, but perhaps even more now that you are more experienced.
So for example, in the AFM study, there are some technical issues that we measure the cell material to cells using colloidal probe microscopy, but we can also use it studying in a different way, like using the single cell to probe the materials underneath. Not actually a part of this publication 3, but I don't think that we match the level of data size that we obtain with the colloidal probe microscopy. So it can also provide a better understanding about the difference setting and how the data provide an insight to the spheroid formation. And also, since we don't have a lot of experience with the AFM with the cells and analyze the force curve, force distance curve, we can also dig a little bit deeper in there and use, for example, machine learning to analyze and differentiate the contribution of different binding events into to the study also. Yeah.
What in the whole work are you most proud of?
I think the AFM, just the sheer time and effort that put into that is, I think it's a massive challenge for us.
Yeah.
And to our colleagues here also. Yeah. So I'm really proud of that work. We started out thinking that we only spent like 3 months on it, and we ended up like with the publication after 2 years. So
Yeah. I can imagine that. So now I'm on the point of the statement. This dissertation involves methods to the use of plant-based components in hydrogels to control the mechanical properties of them and to study their interaction with living cells. So novel methods have been developed, like the AFM force microscope. A new insight in the topic has been obtained. So this has been documented in 3 publications in journals with high impact factor. And those 3 separate publications have been very well connected in the thesis. So new findings have been presented. The author has presented her achievements, pure achievements, clearly and scientifically. So we have had, I would say, very good discussions here today. So you have demonstrated a high level of skills and insights into this field of science. So based on my evaluation of the dissertation and this public defense, I recommend that the doctoral candidate be awarded the grade of pass.
I hereby asking if the audience have any kind of any observation for this doctoral dissertation. Please kindly ask the floor from the custom.
And I'd like to remind you that if you intend to make a written comment about this dissertation, you should tell about this here and now. And official comments about the defense must be submitted to the School of Chemical Engineering within 2 weeks of the defense with an official letter. There seems to be no comments. So then, honored Professor Kristin Syverud, I thank you very much for your great work that you have done today. I mean, or weeks before, probably, because you were so well prepared. You had to spend a lot of time. So I'm really grateful that you took the time to really dig into the Helmi's thesis, and I think you had a very nice discussion today. So with this, I thank you so much, and I declare this dissertation proceeding concluded. Thank you.